This is the final part, the third topic of this chapter. All right. The third topic of this chapter, revision notes. Embryo and its development, post-fertilization events. The development of endosperm and embryo, the maturation of ovule or ovules into seeds, and the ovary into fruit are post-fertilization event. Still it's getting stuck. Okay. Endosperm development. The primary endosperm cell divides repeatedly by mitosis. Primary endosperm cell divides repeatedly by mitosis to form triploid endosperm tissue. Endosperm cells are filled with reserve food material that are used for nutrition of the developing embryo. During endosperm development, the primary endosperm nucleus undergoes successive mitotic nuclear division to give rise to free nuclei. This stage is called free nuclear endosperm. Then the endosperm becomes cellular due to the cell wall formation. For example, the tender coconut water is a free nuclear endosperm that is made up of thousands of nuclei. The surrounding white kernel is called the cellular endosperm. Then the embryo development. The embryo develops at the micropylar end of the embryo sac, where the zygote is situated. The zygote divides only after the formation of certain amount of endosperm to provide nutrition to the developing embryo. The development of embryo is similar in monocotyledons and dicotyledons up to octant stage, 8 divisions. The zygote gives rise to pro-embryo and subsequently to globular, heart-shaped, and mature embryo. We'll be seeing those pictures also. Right? Dicotyledon embryo. It has a central embryonal axis and 2 lateral cotyledons. The portion of the embryonal axis above the level of cotyledons is epicotyle, which terminates into femule, stem tip. The cylindrical portion below the cotyledon is hypocotyle that terminates into radicule, root tip. The root tip is covered with root cap. See here, degenerating synergids, zygote. This is antipodals, primary endosperm nucleus, pen, 3M. All right, already 2 polar nuclei and 1 from the pollen tube, forming PEC, primary endosperm cell. Right? Then zygote starts getting developed and slowly it becomes heart-shaped. Then finally it divides, matured embryo divides into radicle and cotyledons. Femule. Femule is the shoot system. Radicle is the root system. It forms the root system. See here, femule, 2 cotyledons. This is epicotyle. This is hypocotyle, which forms the root system, root cap. What about this monocotyledon embryo? Monocotyledon embryo, they possess only one cotyledon. In the grass family, the cotyledon is called scutellum, which is situated later in the embryonal axis. At the lower end of the embryonal axis, as the radicle and the root cap enclosed in the undifferentiated sheath called coleoreas. The portion of embryonal axis above the level of attachment of the scutellum is the epicotyle, where it forms the shoot system. It has a shoot apex and few leaf primordia enclosed in the hollow folia structure called coleoptile. Next, seeds. Seed is the final product of sexual reproduction. It is the fertilized ovule formed inside fruit. It consists of seed coats, cotyledons, and embryonal axis. The cotyledons are simple, thick, and swollen due to storage of food, as seen in most of the dicots. Mature seeds may be non-aluminous and aluminous. We will see through what it is, don't worry. Embryo of grass. See here, scutellum, coleoptile. This is the shoot system, shoot apex, which starts with the leaves, primary leaves. Then epiblast, radicle, root cap, and coleorias. Then regarding maize, seed, there is a pericarp outside covering, then aileron layer, then endosperm, this is endosperm, scutellum, which has coleoptile, femule, and radicle, right? Maize seed. Then this is monocot seed of onion. See this? There is a seed coat, there is an endosperm, shoot apical meristem, which will start the leaves, which will give rise to that juicy, succulent leaf. Then root tip is there. LS of monocot seed of onion. Non-albuminous or non-endospermic seeds. These seeds have no residual endosperm. If it doesn't have endosperm, residual endosperm, that is, endosperm is completely consumed for embryo development. Example, pea, groundnut, beans. Aluminous or endospermic seeds. These seeds retain a part of endosperm as it is not completely used during the embryo development. Example, wheat, maize, barley, castor, coconut, sunflower. In some seeds like black pepper, beet, the remnants of nucellus also persistent. It is called perisperm. Integments of ovules harden as tough protective seed coat. If you have seen the husk of the paddy husk, you will find that protective layer, integments too. It has a small pore, micropyle, through which oxygen, water enter into the seed during germination. As the seed matures, its water content gets reduced and the seed becomes dry. That is 10 to 15% moisture by mass. The general metabolic activity of the embryo slows down. The embryo may enter the state of inactivity, dormancy. In favorable conditions are available, such as adequate moisture, oxygen, and suitable temperature, they germinate. So the state of dormancy, in temples, in old Hindu temples, there is something known as kalasam at the top, all right, which will be having small pots, inverted pots at the top of the tower, temple tower, all right. Every 12 years, they will replace this kumbha with seeds. Actually, it contains the seeds for germination. This system was developed by the earlier kings. Because of floods and famines and anything, there may be a situation that we won't be having any seeds for cultivation, agriculture. So they keep all the seeds, important seeds in this pot and put it in the top of the tower. If there is a famine, there is no option for germinating seeds, then they can use this seed for germination to again cultivate more and more. That is to maintain that agricultural activity. All right. So it may enter into dormancy, and these seeds can be used for future cultivation. Its life is around 12 years. That's why every 12 years they will be saying that they are having a Kumbh Mela. They used to say Kumbh Mela. All right, Kumbh Mela. Actually, it is scientific. It's not only religious. Actually, it is scientific. Fruits. All right, fruits. The ovary develops into fruit after pollination and fertilization. The transformation of ovules into seeds and ovary into fruit proceeds simultaneously. The wall of the ovary develops into a pericarp. The fruit may be fleshy, as seen in guava, orange, mango, etc., or may be dry, as seen in groundnut, mustard, etc. Many fruits have mechanisms for disposal of seeds. Definitely, disposal of seed is very, very important. Why it is important? When the seed is disposed properly and it gets the desired condition of water, air, and soil, proper soil, rich soil, automatically it forms plants. So the final stage, dispersal of seed, is also important. Fruits are of two types, namely true fruit, where the fruit develops only from the ovary, and other floral parts degenerate and fall off. They are called true fruit. Example, mango, maize, grapes. Then what are false fruit? When parts of flower other than ovary also contribute to fruit formation, they are called false fruit. Apple, strawberry, cashew are all false fruits. That is, the edible part which we eat in apple is made up of thalamus, right? In some species, such as banana, the fruit developed without fertilization. These are called parthenocarpic fruits. Parthenocarpy can be induced through the application of growth hormones. Such fruits are seedless. Advantage of seeds. The pollination and fertilization process are independent of water, while the seed formation is more dependable. Seeds have better adaptive strategies for dispersal to new habitat and help the species to colonize in other areas. They have food reserves, and so young seedlings are nourished until they are capable of photosynthesis. The hard seed coat protects the young embryo. Since seeds are products of sexual reproduction, they generate new genetic combination, leading to variations. The dehydration and dormancy of mature seeds are crucial in storage of seeds. We spoke about that Kumbh Mela Center, that is storage of seeds in case of very, very bad famines. It can be used as food throughout the year and also to raise the crop for the next season. Viability of seeds. Viability of seeds. In a few species, the seed lose viability within few months or live for several years. Some seeds remain alive for hundreds of years. The oldest is lupine, Lupinus arcticus, excavated from Arctic tundra. The seed germinated and flowered after an estimate of 10,000 years of dormancy. 2,000-year-old viable seed is of the date palm, Phoenix dactylifera, dates that dates, discovered during the archaeological excavation of King Herod's place near Dead Sea. See, viability of seed is also important. Some seeds live for several years, some for several months, and some within short period of time. It will be dead. All right. It can't produce the plants. Development of apomictic seeds. We are going to complete the lesson. We are almost at the end of the lesson. In some species, the diploid egg cell is formed without reduction division and develops into embryo without fertilization. In species like citrus and mango varieties, some of the nucellar cells surrounding the embryo sac divide and protrude into the embryo sac. and develop into embryos. Hence, in these species, each ovule contains many embryos. Importance of apomixis in hybrid seed industry. Hybrid seeds have to be produced every year. If the seed collected from hybrids are sown, the plant is the progeny will segregate and lose hybrid characters. The production of hybrid seed is costly. Hence, cost of hybrid seed is also expensive for the farmers. If the hybrids are made into apomin, there is no segregation of characters in the hybrid progeny. This helps farmers to use hybrid seeds to raise new crop year after year without losing hybrid characters. See, this is made up of thalamus. This is made up of thalamus, the edible part of the apple. Check here, thalamus, seeds, endocarp. This is mesocarp in lemon and everything, all right? So these are the sections of apple. If we cut it like this, we'll be having like this. This is apple. When we cut it in the equator, we'll be forming this one. Check how it is formed. So this is the end of the whole second chapter. Sister, did you understand the concept?
Yes, sir.
You had to do a revision. 1 minute.